The purpose of this lab was to examine the effects of adenosine, ADH, and histamine on the flow of fluid through blood vessels and across capillary walls in a frog. Flow rate was measured by counting the number of drips off the frog's toes, and changes in weight due to fluid accumulation in the tissues was measured with an isometric force transducer. The effects on flow rate and weight can be explained by Ohm's law and Starling's equation. Ohm's law describes how the flow of fluid in a tube, Q, is affected by the driving pressure, delta P, and the resistance to flow, R. Resistance in this equation can be expanded, where R equals 8 times nu times L over R to the fourth times pi. Nu refers to fluid viscosity, L is equal to the length of the tube, and R is the radius of the tube. In our case, the tube corresponds to the blood vessels of the frog, and Ohm's law allows us to connect changes in drip rate to physiological changes in pressure and resistance. Starling's equation describes the changes in weight due to fluid movement across the capillary walls. K is the filtration coefficient. Pc refers to the hydrostatic pressure in the capillary pushing fluid across the capillary wall, and pi is the interstitial tissue osmotic pressure, which draws fluid into the tissue. Pi is the interstitial tissue hydrostatic pressure, and Pi-C is the osmotic pressure in the capillaries. These terms affect phi, the flux of water across the capillary wall. When phi is positive, net flux across the capillary wall is directed into the interstitial tissue from the capillaries, reflected as an increase in weight. If phi is negative, the fluid flux is directed out of the interstitial tissue and into the capillary vessels, reflected as a decrease in weight because the fluid drips out of the system and off the frog's toes. To set up this experiment, a dead frog was dissected by taking the skin off the legs, opening up the chest cavity, and taking out everything except the heart, kidneys, and lungs. We accidentally took out the lungs anyway. A catheter was inserted into the frog aorta so that we could perfuse the frog with different solutions. The perfusion reservoir was set at the same height for the duration of the experiment.
Adenosine is a purine nucleoside that is important for biochemical processes, including regulation of blood flow. It has the ability to cause vasoconstriction or vasodilation through receptors on smooth muscle. When adenosine in Ringer's and dextrin solution was perfused through a frog, we saw an increase in drip rate and an increase in weight. According to our results, adenosine acted on the A2A receptor in the smooth muscle of the arterioles via the GS pathway. Activation of the GS pathway increases adenylate cyclase activity, which increases conversion of ATP to CAMP. CAMP activates protein kinase A. pKa causes a conformational change in the potassium channels on the membrane, leading to increased potassium permeability. The increased permeability causes hyperpolarization, which in turn closes voltage-gated calcium channels. That decreases intracellular calcium. At lower concentration, calcium no longer binds to calmodulin, which is an activator of myosin light chain kinase. In smooth muscle, MLCK phosphorylates the myosin, activating it for cross-bridge formation and mechanical contraction. PK also inactivates MLCK, preventing the phosphorylation of myosin, further preventing contraction. Lastly, PK also activates MLCP, myosin light chain phosphatase, which converts phosphorylated myosin back to its inactive form. The decrease in intracellular calcium, the inactivation of MLCK, and the activation of MLCP all decreases contraction in the smooth muscle. This decrease in contraction corresponds to the vasodilation of the smooth muscle, specifically the arterioles in the frog. As per Ohm's law, vasodilation causes an increase in radius, which results in a decreased resistance. As the resistance decreases, flow increases, as shown by the increase in drip rate from 20 to 30 drops per minute. In addition, according to Starling's equation, the precapillary vasodilation in the arterioles allows more blood flow in the capillaries, causing PC to increase. The increased pressure in the capillaries pushes fluid across the capillary wall into the interstitial tissue. An increase in flux is reflected by the increased weight from negative 0.03 to 0.08 grams per minute.
Antidiuretic hormone, also known as vasopressin, can cause vasoconstriction via binding to the V1 receptors in the smooth muscle of the arterioles. V1 receptors are GQ protein-coupled receptors. When vasopressin binds to the V1 receptor, the GQ protein activates phospholipase C, PLC, causing PIP2 in the membrane to be cleaved into IP3 and membrane-bound diacylglycerol, DAG. IP3 increases the release of calcium from the sarcoplasmic reticulum by binding to the IP3-sensitive calcium channel, IP3R. Meanwhile, diacylglycerol activates protein kinase C, PKC, which activates L-type calcium channels, LTCCs, in the membrane. This increases the influx of calcium into the muscle cell from the extracellular fluid. This increase in calcium both from the SR and the extracellular fluid allows for calmodulin, CAM, to more often be bound by 4 calcium ions. This activates MLCK, which converts myosin ATPase into its active form, where it can convert ATP to ADP. This in turn allows for phosphorylation of the myosin head from its inactive form to its phosphorylated active form, allowing myosin to form cross bridges with actin and initiate a power stroke to cause contraction. PKC also acts to decrease the activation of potassium channels, such that potassium does not leave the cells readily, increasing the length of depolarization and allowing the LTCCs to stay open longer, resulting in a larger influx of calcium. The effects of IP3 on the SR and diacylglycerol on the LTCCs and potassium channels all allow for greater contraction of the smooth muscle. The net effect is vasoconstriction in the arterioles. This vasoconstriction affects both the flow of fluid through the blood vessels as well as across the capillary walls. In terms of Ohm's law, this vasoconstriction decreases the radius of the arterioles, increasing the resistance and therefore decreasing the flow. This decrease in flow is reflected in the changes in drip rate from 32 drops per minute to 30 drops per minute. As well, the decreased precapillary flow through the arterioles decreases the capillary pressure, PC. This decreases phi, and the relative net flux of fluid is directed into the capillaries out of the tissue. As the fluid exits into the capillaries and flows through the vasculature of the frog, it eventually leaves the frog at the end of the systemic circulation, which is reflected as a decrease in weight from 0.04 to 0.03 grams per minute.
Histamine is a biogenic monoamine involved in inflammatory responses through activation of G protein-coupled receptors. In our experiment, we added histamine in Ringer's and dextrin solution and observed a decrease in flow rate and weight. When histamine binds to H1 receptors in smooth muscle, an intracellular signaling cascade is triggered. Like ADH on V1 receptors, histamine's H1 receptor has a GQ protein-coupled pathway that stimulates PLC, phospholipase C, to convert PIP2 into diacylglycerol and IP3. Diacylglycerol then stimulates protein kinase C to activate L-type calcium channels in the cell membrane. PKC also inhibits potassium channels. These two effects of PKC allow for influx of calcium ions and prolonged depolarization of the cell. The other product of PIP2 conversion, IP3, goes on to activate IP3 receptors in the membrane of the SR, a membrane-bound organelle responsible for calcium handling in smooth muscle cells. The activation of IP3 receptors allows for increased calcium release from the SR, which allows more calcium to enter the cytoplasm. Once in the cytoplasm, calcium activates calmodulin, which allows myosin light chain kinase, MLCK, to convert the inactive form of myosin ATPase into the phosphorylated active form. Activation of myosin ATPase converts ATP to ADP, allowing the myosin heads to be phosphorylated and to power muscle contraction. In this way, the binding of histamine to H1 receptors allows for the contraction of blood vessels. Given this understanding of how histamine acts to increase vasoconstriction in this experiment, the decrease in flow rate observed in our data can be explained by Ohm's law. Here, flow rate decreased from 30 to 26 drops per minute. Vasoconstriction decreases the radius of blood vessels, and because Ohm's law shows that radius and resistance are inversely proportional, any decrease in radius will result in an increase in resistance. Because resistance is inversely proportional to fluid flow, any increase in resistance causes a decrease in fluid flow. Decreased fluid flow rate was observed in our drip rate data, which means it is supported by the physiology described. The data from this portion of the experiment also showed a decrease in weight, which can be described by Starling's equation. When blood vessels vasoconstrict, the resulting increased resistance in the arterioles causes a decrease in flow rate. This means that less fluid will be transported through the capillaries of unchanged size. Less fluid occupying a container of unchanged volume equates to a decrease in pressure. In this case, a decrease in capillary pressure, PC. Decreased PC means phi is decreased, and the observed decrease in weight is shown by the data. The weight decreased from negative 0.03 to negative 0.05 grams per minute.
Overall, adenosine's effect on the A2A GS protein-coupled receptors caused an increase in drip rate and in the weight of rate gain through vasodilation in the arterioles. ADH on V1 receptors and histamine on H1 receptors, both coupled to GQ pathways, resulted in a decrease in drip rate and weight as the result of vasoconstriction.